Hello everyone, my name is Jamshed Khan and I'm a PhD student in computer science at the University of Maryland College Park, working with Dr. Rob Patro. I'll be presenting our recent research work titled as Cuttlefish, Fast Parallel and Low Memory Compaction of de Bruijn graphs from Large-Scale Genome Collections, authored by me and Rob and accepted for ISMB 2021. To provide some context behind the research, with modern high-throughput sequencing, today we have thousands of assembled genome sequences, up to mammalian sized. We also have successfully assembled genomes that are orders of magnitude larger than the typical mammalian genome. As a result, there is an increasing need of efficient sequence indices to align short and long reads to these reference sequences. We also need efficient representational data structures for these references to perform comparative genomics and pangenome analysis. Thus, compact representations of whole genome reference collections are of acute interest in bioinformatics today. So, to represent some collection of references, the simplest way is to have each reference as just a string of constituent nucleobases. In this example figure, we have two strings, the blue one and the red one, each representing a genome reference. The blocks here denote identical substrings. For example, the green block is present twice across the genome, whereas this purple block is present in 3 copies across the genome. What we may instead want to have is something like this, where the repeated blocks are unified. Besides providing space savings, such a representation also aids in having efficient indices over these references and easier comparison across them. An excellent object to represent such genome references and also sequencing reads is the de Bruijn graph. Historically, de Bruijn graphs have been highly utilized in genome assembly and are increasingly being used in comparative genomics and sequence indexing. And the colored and compacted variant is of specific interest in this research. In this example, we see two de Bruijn graphs, each one constructed over a separate reference sequence. The graph at the top corresponds to the red sequence and the one at the bottom to the blue sequence. We are using k equals 2 here. Each distinct k-mer, that is substring of length k of an input sequence, corresponds to a unique vertex in a de Bruijn graph. When having a single de Bruijn graph over this collection of two reference sequences, we unify the repeated vertices across the graph into single vertices in the union graph. For example, for the two de Bruijn graphs mentioned earlier, the 4 repeated pairs of vertices get unified into 4 single vertices in this union de Bruijn graph at the bottom. In this paper, we tackle the initial steps of de Bruijn graph-based genome analysis pipelines, which are the construction of the colored and compacted de Bruijn graph for reference collections in time and memory efficient manner. To provide some preliminary technical details, we use the bidirected variant of de Bruijn graph in this work. Each vertex in a bidirected de Bruijn graph corresponds to a canonical k-mer, instead of just an ordinary k-mer. That is, each vertex has two associated k-mers. One is the canonical k-mer and the other is its reverse complement form. For example, in this de Bruijn graph, the vertex labeled with CGA corresponds to two k-mer forms. One is the CGA form and the other is its reverse complement form. So, what is the compacted variant of an ordinary de Bruijn graph? When you collapse each maximal non-branching path of a graph into a single vertex, the resulting contracted graph is called the compacted variant of the original graph. For example, in this ordinary de Bruijn graph, you can see 4 non-branching paths that are maximal. That is, the path cannot be extended towards any direction anymore. Collapsing each of these paths into single vertices provides you with this contracted graph GC in the right, which is the compacted de Bruijn graph for the input sequence collection S, and with k equals 3. You'll also note that these paths are completely disjoint. Given a set R of whole genome references, a naive algorithm to construct the compacted de Bruijn graph over R may go like this. First, construct the ordinary de Bruijn graph G by making a linear scan over the sequences in R and storing the adjacency information along the way is some data structure like an adjacency matrix or an adjacency list. Then, using some linear graph traversal algorithm like the depth first search or the breadth first search over G, you can enumerate all the maximal non-branching paths in one go, which provides you with the compacted graph GC. Now, this approach can quickly become infeasible for moderate to large-sized genomes due to the enormous space requirements of the ordinary de Bruijn graph G. Hence, what we need is some sort of construction algorithm for GC that may bypass an explicit construction of G. A motivating observation aiding in bypassing the construction of the original graph G is that you may obtain an implicit walkover G without having G explicitly in hand. So consider a De Bruijn graph G over a single input string S. When using an edge-centric De Bruijn graph, a complete walk traversal W can be obtained over G by making a linear scan over S. Each k-mer seen along the scan is a vertex in G, and adjacent k-mers in the string are neighbors in the graph, providing you a complete walk over G. And this might also be observed that each non-branching path, also referred to as unitigs, must be present as subpaths in such complete walks. This observation is proven in the paper and provides us with the insight that by making an implicit walk over G, we will encounter each maximal unitig along the way, providing you with all the vertices of the compacted graph. An important notion for the maximal unitigs is their flanking vertices. If each vertex in G can be characterized as either flanking or internal for their containing maximal unitigs, the unitigs themselves can then be extracted through identifying subpaths in the input strings that have flanking vertices at both their ends. This way, the graph compaction problem is reduced to the problem of determining a set of all the flanking vertices in the graph. A vertex V is called the flanking vertex if it has at least one side SV such that SV either has zero incident edge or more than one edges, or SV has exactly one incident edge that connects V to some other vertex U and to its side SU, but SU has multiple incident edges. For example, consider the vertex CGA here. Its front side has zero edges, and that makes it a flanking vertex. For the vertex CTC here, both its sides has a single unique edge, but one of its sides is connected to this side of AGA that has multiple incident edges at that side, which makes CTC a flanking vertex. This way, you may enumerate all the flanking vertices if you have the graph at hand. Now, it is important to note that for the purposes of enumerating the flanking vertices, not all the adjacent information of all the vertices need be tracked. We can throw away much of this information at no cost. A side SV of a vertex V can be considered to be in one of 5 different configurations in the graph. One configuration for each possible unique singleton edge, and there are 4 possibilities for such, and one for when the side does not have one unique incident edge. And since the vertex has 2 sides, as a whole, a vertex can be in 5 times 5, that is 25 different configurations. We refer to each of these configurations as a state. So in this paper, we propose a deterministic finite state automaton, or a DFA model, for each vertex. Each vertex in the graph is treated as a separate DFA. A DFA is formally described with 5 parameters, and we briefly gloss over 3 of those here. Q0 is the initial state of each vertex, that is, this is referred to as the unvisited state. Before making any graph traversal, we do not have any information on any of the vertices yet, so each DFA in the graph starts from this state. Q prime is the set of all the possible 25 different states for the DFA. These 25 states can be partitioned into 4 disjoint classes based on their structural characteristics of the associated vertices in the graph. For example, for a vertex that has a unique edge at the front and a unique edge at the back, you can have 4 times 4, that is 16 different configurations for that vertex. These 16 configurations of states are bundled into this one class named single in, single out. Similarly, you can have 4 different states for some vertex that has a unique edge only in its back, and these states are said to be in this multi-in, single out class. Delta is the function that governs the state transition for each DFA during the graph traversal, and this is the state transition diagram for the automata. We are not going to go into much details into this due to time constraints, This is a schematic diagram detailing various transition scenarios that the different automata in the graph may follow during the lifetime of the algorithm. You can find the figure in the supplementary materials of the paper. So, since each vertex, that is each canonical k-mer, has a transitioning state associated with it, we need some sort of associative data structure for the canonical k-mers and their states. For such, we design a hash table. As the hash function, we use a minimum perfect hash, which works correctly here since the set of keys, that is, the set of canonical k-mers, is static. We use the BB hash algorithm to construct the hash function that needs just around 3.7 bits per distinct k-mer to store the function. As for the hash table buckets that actually store the states of the vertices, we use a bit packed array. Now, since a vertex always remains in one of only 26 different states, we require log26 base 2, that is 5 bits to store such a state. Thus, as a whole, the hash table structure requires approximately 8.7 bits per distinct canonical k-mer. So, given a set S of reference sequences, so the overall algorithm goes as follows. Firstly, the set K of unique canonical k-mers is extracted using the k-mer counting tool KMC3. Then, a minimum perfect hash function H is constructed over the set of keys K using the BB hash algorithm. Then, making an implicit traversal over the underlying de Bruijn graph and making appropriate state transitions for the vertices encountered along the way, the actual state of each vertex of the graph is computed. Then, once we have all the states information for all the vertices, another implicit traversal over the graph is made similarly, but this time using the available states information, the vertices encountered along the way are characterized as either flanking or not for their containing maximum unitics, and the maximum unitics are extracted on the fly. To assess the performance of the algorithm on real data, we used a varied collection of datasets of different characteristics. As single input genomes, we used two mammalian references, the human and the western gorilla genomes, and also the quite large sugar pine genome. For multiple input references, we used a variety of datasets, 62 small bacterial genomes from E. coli, 7 humans and 7 apes as a moderate collection of mammalian genomes, and two quite large reference collections, which are 11 diverse conifer plant references and 100 human references. So this here is quite a busy table showing the performance of Cuttlefish in single input graph compaction compared against other state-of-the-art tools for whole genome reference compaction algorithms. We compared the performance against Bifrost, DGSM, and Tobacco. Among these, both Tobacco and Cuttlefish are applicable only for whole genome references, whereas Bifrost and DGSM both are applicable also for sequencing reads. Now, let us try to parse two rows from this table. For the human genome, when using 8 threads and K-code61, Bifrost builds the compacted graph in an hour and 20 minutes using 50 gigabytes of RAM. DGSM takes around 47 minutes using 84 gigabytes of RAM. Tobacco takes just around 11 minutes and using 5.5 gigs of RAM, whereas Cuttlefish builds the graph in 7 minutes and 45 seconds and using around 3 gigabytes of RAM. For the large sugar pine genome reference and using 16 threads, when you're using K-code31, Bifrost took around 22 hours and 229 gigabytes of memory. DGSM took 9 hours and half using 145 gigs of memory. Tobacco took an hour and 50 minutes using 62 gigs of memory, whereas Cuttlefish took just around 51 minutes using 14 gigs of memory. So the highlighted values in each row depict the best running time and memory usage. Similarly, for multiple input strings, Cuttlefish performed better than the other two. For example, for the 7 humans dataset using 16 threads, Cuttlefish required 21 minutes using just shy of 3 gigs of memory, while the other tools required from at least 30 minutes to up to 95 minutes, with at least 6 gigabytes of memory and up to 38 gigabytes of memory. For 100 humans, we were able to run only Tobacco and Cuttlefish in our hardware, with Tobacco requiring around 23 hours and 126 gigs of memory, while Cuttlefish required less than 9 hours and using just 29 gigabytes of memory. We show the timing performance of the algorithm here. When applied with an increasing number of threads, we see that the running time of all the 4 steps of the algorithm drop with an increasing number of processor threads. A better demonstration for this is actually the speed up plot for the steps. So this is the parallel speed up plot for the various steps. We see that the initial k-mer set extraction scale roughly up to 8 threads and saturating afterwards. The minimal perfect hash function construction scales up to around 24 threads. The last two steps that involve traversing the graph scale almost nearly all the way up to 32 threads. So through the novel idea of modeling a de Bruijn graph vertex as a DFA, we tried to push the boundary of our ability to construct compacted de Bruijn graph vertex construction, both in terms of genome scale and count, which we hope would facilitate further improvements in the role of de Bruijn graph in genomics. Cuttlefish is available in GitHub under an open source license, and it is also available in Bioconda. And lastly, thank you everyone for attending the presentation. I'll be happy to answer your queries and also discuss further prospects for Cuttlefish with you. Regards.